Hi, Jessie. It's so nice to have you here today to have our Inflammation, Fibrosis, and Cancer Journal Club. For those of you who do not know, Dr. Jessie Su is a researcher at Accutar Biotechnology, and she's going to speak to us today about TGF-β. Thank you so much for being here, Jessie.
Thank you for inviting me, Dr. Salvatore. And I'm going to share my screen, and today I'll talk about my research when I did my postdoc at Memorial Sloan Kettering. TGF-β orchestrates fibrogenic and developmental EMTs with the RAS effector RREB1. And a brief introduction of myself. I did my Ph.D. study at Mount Sinai in New York. And in my Ph.D. work, I focus on studying the pluripotency of embryonic stem cells. And we found an important factor to maintain ESL pluripotency. And also the second project, we used the induced pluripotent stem cell as a model to study Li-Fraumeni syndrome. And after PhD, I changed my direction of research to cancer research. And I did my postdoc study at Memorial Sloan Kettering Cancer Center. And over there, I did the study TGF-β orchestrates fibrogenic and developmental EMTs with the RAS effector RREB1. And currently, I move my career into the biotech company. Currently, I'm doing research work at Accutar Biotechnology, focusing on developing new types of degraders and small molecule inhibitors for cancer target therapy. And a brief wrap up of my PhD work. So in my PhD, the first project is studying the pluripotency of ES cells. So to study that, we did a shRNA screening of hundreds of kinases and phosphatases. And we found that Aurora kinase A is important for maintaining ES cell pluripotency through phosphorylation and inactivation of p53. So when there is no Aurora kinase A, the stem cell would undergo differentiation and lose the pluripotency. That's because there's no Aurora A and there's no phosphorylation of p53. And p53 combined to the differentiation promoter genes and induce the differentiation genes. And when there is Aurora A, it can phosphorylate and inactivate p53. So the cells is pluripotent. And when we induce the fibroblast to iPSCs, p53 is not interfering this process, and Aurora A is promote this process. So for this paper, we submitted a cover for cell stem cell where it was published, and p53 is considered the super tumor suppressor genes that has multiple functions. So we think it's p53 is Superman. And in this context, Aurora A, which is a polar light, and Superman is afraid of Aurora A, the lights. And by inactivating the Superman, the stem cell can be maintained in pluripotent stage and not induced by the Superman to differentiation.
Very exciting work. So exciting, so important.
Yeah, so this is like power struggles in ES cell fate. ES cell can be stem cell and ES cell can differentiate in all kinds of other cells.
Amazing.
That's the first part of my PhD. Then second part, we are very encouraged by this new model called iPSC. We think it's a model we can use to study for those diseases we have no model to study before. So Li-Fraumeni syndrome patients, they are the patients has inherited p53 mutation or deletions. And this patient can develop multiple types of cancer at a very early stage. And a lot of the cancers are pediatric cancers, for example, osteosarcoma. So in this project, we get the skin biopsy, skin fibroblast from many patients. And these patients have osteosarcoma. We use these fibroblasts and Yamanaka factors to induce this fibroblast back to the stem cell state. So this is called patient-derived iPSCs. And use the stem cell, patient-derived iPSCs, we can differentiate this stem cell to specific lineage. In our case, our patients have osteosarcoma, so we induce these iPSCs to mesenchymal stem cells, then osteo lineages, osteoclasts and osteoblasts, and looking at the process of this osteo lineage differentiation, we can compare the leaf-from-many patients iPSCs with the normal iPSCs and to see what problem does this patient have during development. And osteosarcoma is a cancer type that related with abnormal bone development. And to compare the patients and normal, we found out important factors in this process. And we try to use the technology of CRISPR to see if we can correct the mutation and also correct the phenotype. So here we are very exciting that we use both technology that got a Nobel Prize. One is Yamanaka's iPSC and the other is CRISPR gene editing. So that's a brief wrap up of my PhD work. So I will focus on discussing my postdoc work that about TGF-β signaling. So first, a brief introduction. In the TGF-β signaling project, I started with a pancreatic cancer model. Pancreatic cancer is a very extremely devastating disease that we don't have much treatment right now. And with all the development in cancer research, by 2030, pancreatic cancer is projected to become the second most cancer deaths in the US by 2030. And in the development of tumorigenesis of pancreatic adenocarcinoma, there are different stages. And based on current understanding and research, we think that different mutations are involved in different stages. In the beginning of panel 1, panel 1A and 1B, there's usually accompanied with KRAS mutation. And in the later stage, we notice the patient have P53 mutation and SMAD4 mutation. So over 90% of the patient have KRAS mutation. I feel like this like the first cancer driver, KRAS, it sets the cells in a specific state that is more fragile for the second mutation. And we see that the second mutation, a lot of players in TGF-β signaling are mutated in pancreatic cancer, including SMAD4 happens in almost half of the patient, and TGF-β receptor 1, TGF-β receptor 2, together with SMAD2 and SMAD3, TGF-β signaling pathway is mutated in more than half of pancreatic cancer patients. And a brief introduction of TGF-β signaling pathway. So TGF-β signaling pathway is triggered by TGF-β, which is a ligand. And the ligand can bind to TGF-β receptor 1 and TGF-β receptor 2. And the receptor 1 and 2 can form a dimer, heterodimer, And this dimer is activated and it can phosphorylate RSMAT, which is usually SMAT2, SMAT3. SMAT2, SMAT3, after its phosphorylation, it can locate from the cytoplasm to nucleus. And in nucleus, SMAT2, SMAT3 can bind to SMAT4 or TRIM33. Together, this SMAT2, 3, SMAT4 trimer can induce downstream transcriptional regulation. And in different contexts, TGF-β can have different function. So it has very diverse function that is very fascinating and it's everywhere. So in the early stage, TGF-β, for stem cell, TGF-β can induce differentiation, usually mesoderm and ectoderm. And also TGF-β has function in regeneration. And in the early stage of cancer, we think TGF-β is a tumor suppressor. And in the late stage of cancer, we think TGF-β is associated with tumor progression and metastasis. I think this function is majorly related with EMT. So TGF-β can induce EMT. TGF-β have multiple functions and EMT also have multifunctions. I will discuss this later in detail. So how does TGF-β has multiple functions? The same signaling pathways has multiple functions in different contexts. We think that in different cell contexts, the cell type, different cell types in different tissues, like in different stage, in different condition, like healthy or cancer, stem cell, pancreatic cells, in different cell types, different cell contexts, the chromatin of the cells are open in different stage. Like the chromatin state is different. And the activated transcript factor are different in different cell types. For example, in cell type A, the orange transcription factor is activated. And the TGF-β, when it needs to transduce some gene and when need to carry on some function and need to bind with some other factor to do this together. So the activated SMAT2, 3, when it comes into nucleus, and need to look for the open area of the chromatin and need to look for the active partner of the SMATs. We call this LDTFs, lineage determining transcription factors. And for example, in cell type 1, SMAT can bind to the orange partner and transcribe the gene A, B, and C. And in cell type 2, because the chromatin stage is different and the green factor is active, SMAT can find the green factor and transcribe the gene DEF. That we think is a theory to explain why TGF-β have so many different functions in different contexts, like development, fibrosis, cancer, early stage of cancer, late stage of cancer.
I see. That's how it's modified.
Yeah. So here we ask two major questions. What is the context to TGF-β cofactor in the early stage pancreatic cancer? And what is the cofactor function in different contexts? That's basically the two questions for this project. And first, I use mutant KRAS cells to start this project. So we have the transgenic mice, and I isolated these pancreatic epithelial cells and put them in organoid culture, and I induce TGF-β G12D mutation in this organoid. And we can see that this is TGF-β inhibitor. This is TGF-β. So we see that with KRAS on, TGF-β can induce EMT in this organoid. So organoid is a 3D culture. When EMT happens, the organoid sphere fell apart and become monolayer culture. And it loses the E-cadherin expression and gain ZEB1 expression here. And it only happens in the KRAS G12D organoid, but not in the wild type organoid. That means KRAS can set the cells in specific state. When TGF-β come in, it can induce EMT, robust EMT, while the wild type cells, they don't do that.
What turns KRAS on? Is there a lot of things that turn KRAS on?
So that's a very interesting question. That's like, how do people get cancer?
I'm sorry.
I think yeah, KRAS is an oncogene. Yeah, and we notice that it happens, mutation happens in multiple cancers, like pancreatic cancer is #1, colorectal cancer, a lot, lung cancer, some of the types. It could be different carcinogen or different factors to cause KRAS. But very exciting nowadays, people are trying to develop KRAS inhibitors in the field. It could be applied to a lot of cancer types.
Thank
So
you.
I have a video to show you when the wild type organoid and KRAS mutant organoid treat with TGF-β. And we can see this is the organoid, this sphere, they're like little bubbles in the matrigels. And when there's TGF-β, it loses the 3D structure and become monolayer. And this is EMT, and the cell go apoptosis.
Oh my gosh.
Go apoptosis. Whereas you see the wild type organoid, they're still intact. And I think this is a mechanism for early stage TGF-β tumor suppression. So in the early stage, we have KRAS, but one mutation is not enough to drive cancer. And the TGF-β is a barrier to pass. TGF-β can induce EMT and induce apoptosis in cells acute, bad. And when there's a second pressure, the cell lose TGF-β. There's a mutation there's a non-mutation in SMAD4, there's a mutation in the receptors. The cells lose the protection barrier from TGF-β, and Z can keep growing. They don't undergo EMT and death. This is a special, very special context, one of the function, tumor suppression in the early stage. And we want to use this model to study it. We can see with the TGF-β induction, there's a high expression of SNIL, high expression of SMAD4, but the robust EMT only happens in the KRAS organoid. And we develop another model, similar context, with the SMAD4 deletion, SMAD4 non, and the KRAS mutation. And in this cell, we overexpress SMAD, and they are very sensitive to TGF-β. And the cells undergo EMT and apoptosis. These cells is monolayer cells. They are very easy to use in research. We use it to develop a screening we're going to do next. So another image showing that this is the cells with TGF-β treatment 3 hours. By 36 hours, the cell become from epithelial to mesenchymal. So they're undergoing EMT. And as the time goes, we can see more and more dead cells. So they're undergoing EMT first, then apoptosis. So we can use this model to design a screening that if we knock down a gene that is important for cell death, EMT and cell death, if we knock it down, the cell will survive. you will see more cells survive. And this now shRNA will be enriched after TGF-β treatment. So we developed this screening, transfected the cells with different shRNAs as a pool, and we found there are two candidates. REB1 is one of the candidates in this screen, and SOX4 is another. SOX4, we did another paper, that's a previous paper, and this paper is focused on REB1. And what is REB1? REB1 is RAS responsive element binding protein 1. So it's actually very exciting to get this candidate after our screening because we are interested in RAS context. And REB1 is a RAS responsive gene. And there's not much study and research studying REB1. The only thing I can find back in that time is it is related in RAS signaling, but the function is unknown. It is a very large protein. It has multiple zinc fingers. And REB1 homolog regulates cell adhesion and migration in Drosophila, which sounds promising. Like it might have a function in EMT. And REB1 is downregulated in pancreatic cancer. And the recurrent REB1 somatic mutation is found in pancreatic adenocarcinoma. Based on a new study in TCGA in 2017, it was the first time it was reported in pancreatic cancer. It has 5%, which is low, but it sounds promising to me. It's already the top genes. And so next step, I want to see if REB1 is a specific LDTFs in KRAS mutant cells. So we first did a chip-seq analysis of SMAD2/3 and REB1. So I want to see if REB1 is located close to SMAD2/3. Like SMAD2/3 may bind with REB1 to induce specific gene transcription. So here is overlapping SMAD2/3 chip-seq peaks with REB1 chip peaks. We can see a very interesting thing here. So with SB TGF-β inhibitor treatment, there's no SMAD2/3 binding. It's inactive. With the ligand TGF-β treatment, SMAD2/3 is active, and here is the peak. But the interesting thing is REB1 peaks in both SB and TGF-β condition. That means REB1 is already there, even in the SB treatment condition. And when TGF-β is there, REB1 is still there. That's when we think back the picture of the orange and the green LDTFs. REB1 could be a specific LDTF that is already there in the KRAS cells. And when TGF-β signaling kick in, it might quickly find REB1 and induce the downstream EMT and gene expression. So if we take a closer look, so REB1 wild type, SMAD, this is a SMAD2/3 chip-seq graph. In the wild type REB1 cells, it has the peaks in downstream genes, for example, SNAIL1 and HAS2. And when we knock out REB1 in the cells, there's a much less binding of SMAD2/3. So when there is no REB1, SMAD2/3 is hard, has difficulty for SMAD2/3 to bind at SNAIL1 and HAS2 sites. Similar experiment, we inoculate wild type cells and REB1 knockout cells in sub-Q in mice. We can see that with when there is a REB1, the tumor doesn't grow much. And when there is no REB1, the tumor grows. And we can see that when there is REB1, there's tumor suppression and apoptosis. And when there is no REB1, the tumor, subacute tumor grow very big. And that's in the early stage. That's aligned with the early stage function of TGF-β. It's actually tumor suppression. But in the later stage, I will talk later, it can promote metastasis. So we studied the tumor early stage in pancreatic cancer and what is the REB1 function in different contexts. For example, in the late stage, TGF-β is a metastatic factor. Does REB1 also have a function in there? We're going to see. So here I use another model. It's lung adenocarcinoma cells, and it's a late stage tumor type. And in the wild type without, in the wild type cells with REB1, the tumor grow to a big size. And when we knock out REB1, tumor shrink a lot. And we can see less tumor module in these lungs, less tumor signals. And in the latest stage of tumor, TGF-β promotes the tumor growth and promotes metastasis, promotes fibrosis, and so is REB1. We can see without REB1, there's less tumor growth and less signal. And if we dissect these tumors from the last slide, dissect these tumors, we can see that in the big tumor with wild type REB1, there's more fibrogenic marker expression. And in the REB1 knockout, which has less TGF-β, we can also see less fibrogenic marker expression.
Very interesting.
Yeah, so we think that TGF-β has multiple function, and TGF-β induce EMT. EMT also have multifunction. It could be good, could be bad. In the early stage of the embryo from stem cells, I need to differentiate into all kinds of tissue types, cell types. EMT is very important for this process. And for disease model, fibrosis, EMT can induce fibrosis, and TGF-β over, a lot of TGF-β is considered bad. And in tumor progression, EMT induce metastasis and the migration of the tumor cells. TGF-β is also considered bad. So I want to look as my background for Ph.D. stem cell study research. So I want to look back for the embryo to see whether REB1 and TGF-β play a very important role in the development. So here is a database. We can put in any gene we want and see the expression levels of different genes in this embryo, E7.0 mouse embryo, anterior, posterior, left, and right. And snail 1, goosecoid, brachyury, ADH2, these are the differentiation genes. We can see that snail is highly expressed in the posterior area. And when we put REB1 to search, it also shows a higher expression in the posterior area. Yeah, and so I use stem cell in culture. We remove leaf, which is a potency marker factor. Remove leaf in the stem cell culture can cause the cell undergo differentiation. And in the special plate, it forms a sphere structure. It's called embroid body, EB. And looking like different days, 0, 1, 2, 3, 4 days of EB differentiation. And we can see by day 4, EB day 4, all these genes are upregulated. it's an in vitro model to mimic the differentiation, like day 7 embryo, it has this upregulation of genes, and in the dish, day 4 remove leaf, we can also see upregulation of these genes. And we use these genes as a signature and look at the ES cells, wild type versus RAB1 knockout. So in the wild type, day 4, we can see a nice pattern of upregulation of these differentiation genes. And in RAB1 knockout, day 0 compared to day 4 does not show this differentiation pattern. So the differentiation is impaired by depletion of RAB1. And the EMT markers, ZAP2, snail, uncut herring, snail 2, has 2, this EMT genes is also dysregulated in day 4 RAB1 knockout. That suggests the RAB1 also play a role in embryonic differentiation and embryonic EMT.
Jessie, we're going to run out of time soon. I'm sorry, I just...
So that is the last part. We put the cells into mice and see if the embryo is normal. It is not. The embryo is very abnormal by day 7.5. By day 8.5, when the normal embryo has a head and tail structure, and this knockout RAB1 embryo, it shows very abnormal structure, and some of them even develop 2 heads and 2 tails, or 2 tails. And when we look into the markers, it has the abnormal EMT marker expression, the cut herring and uncut herring. Instead of forming a nice layer, it has a very mixtured expression.
Jessie, your work is so interesting. Amazing.
Thank you so much. And I'm going to wrap up.
Sorry?
I'm going to finish soon, go back to this model that we think in this context, different contexts in ES cells or pancreatic cancer, RAB1 is there, but the RAB1 bind to different sites on the chromatin. And therefore, when SMAD4 is activated, it can find the RAB1, but on different location, and it can transcribe snail and glucoid in ES cells, for example, snail and the WISP1 in the fibrogenic process, for example. And yeah.